Hello! Today I'll be covering the topic of evolution. I'll begin by explaining what evolution is, and then I'll move on to incorporating the labs we covered to help demonstrate the topic. So, what is evolution? The dictionary definition of evolution describes it as anatomical changes that populations of organisms undergo from generation to generation due to the process of natural selection. Let's see an example of this. In the United Kingdom, there is a species of moths called the peppered moths. The Industrial Revolution changed the dominating color of the moths. Before the Industrial Revolution, peppered moths were widely white with black specks to allow it to camouflage on white tree trunks to hide from predators. However, as soot from factories blackened trees, the black moth emerged as a dominating color. Natural selection drove this change of color in the population. There were already black peppered moths before the Industrial Revolution, but their color wasn't favorable since birds could see them easily on the white tree trunks, so the frequency of the black allele in the population wasn't high. As you can see here, during the revolution, the white peppered moth's color was unfavorable since birds could easily spot them on blackened tree trunks, so the frequency of the white allele decreased while the frequency of the black allele increased and were passed on to future generations. Let's look at the labs now. I chose to use content from Musculoskeletal 2 and Musculoskeletal 4 labs to demonstrate the concept of evolution. I chose these labs because both explore the connectivity of bones that interact with the rest of the skeleton and how they react to environmental pressures, indicating evidence of evolution. The bigger picture of these labs is that studying bones from different time periods can show us that evolution has occurred, and their shapes and connectivity with other bones in the skeleton tells us a lot about how the organism lived, letting us learn more about life in the past. In Musculoskeletal 2, we learn about bone remodeling. Bones remodel themselves to adjust to pressures and injuries they experience. Here, you can see the compact bone and trabecular bone. The denser portion on the surface of the bone is compact bone. It's remodeled at the periosteum or endosteum, which is connective tissue on the outer surface of the bone and connective tissue on the inner surface of the bone, respectively. This ability enables it to change the thickness of the outer surface of the bone. On the other hand, trabecular bone, the spongy portion of the bone composed of trabeculae, is remodeled at only the endosteum. This allows it to increase its density to better support the forces applied to the bone. As you can probably guess, bones can tell us a lot about someone's active lifestyle, whether he often lifts heavy weights or not, if he gets injured a lot, or if he isn't active as much. The process of bone remodeling adjusts as we adjust the ways we live. So if you walk and run a lot, it'd be expected that you'd have denser trabeculae than someone else who doesn't walk or run a lot. This thought process can be applied to primates. Early humans traveled by foot a lot more than modern humans do, and as humans walk less as a population, trabeculae density decreases in the population. This image shows that early humans had denser trabeculae than we do now because it would have been more favorable for them since it'd be able to handle the body weight for long periods of time. Hence, as scientists try to answer the question as to why bipedalism came about, studying bone remodeling is crucial as it is one of the biggest indicators of evolution. In Musculoskeletal 4, we watched America's lecture on her research, which allowed us to complete the activity about comparative anatomy with hominids on the next page. In summary, we analyzed the bone structures associated with bipedalism in both chimpanzees and humans. Here's a picture of a chimpanzee skeleton and a human skeleton. The major differences are that chimpanzees have a longer pelvis than humans do, and humans have a wider, more bowl-shaped pelvis. Additionally, chimpanzees' femur is straighter and more linear than that of humans. Humans' femur is angled inwards to the center of the body. This angle is known as the bicondylar angle, which the National Institutes of Health defines as the angle between the shaft of the femur and a line that is perpendicular to the transverse plane of the body. The angle our femur is at allows our feet to be lined up with the center of gravity directly below the trunk, giving us greater stability when we walk, since both legs take turns supporting our body weight. It also makes it a lot more energy efficient for us to walk on two legs. Since chimpanzees' femur isn't angled like ours, it must maintain support from both legs simultaneously. We also briefly analyzed the skeleton of Lucy, widely considered as the first modern human ancestor. Her skeleton, especially her pelvis and femur, strongly resembles that of the modern human. These observations of hominids' skeletons are proof of evolution because over generations of early humans, many skeletal changes occurred as their lifestyles and the environment changed that made them eventually look like the modern human. Bone structures allow us to formulate ideas of when bipedal humans first evolved, which can lead us to study the factors that pressured bipedalism and humans to evolve to better adapt to their environment. That's all I have for today, and I hope you had as much fun learning about evolution as I did. Thanks for watching.